I'm going to be talking about several different issues which I think are critical in thinking about this series of disorders. And I say series of disorders because obviously there's a wide spectrum of disease and symptoms and such. I think it's important nonetheless that you hold together, because if you begin to parse it and think in terms of people with this specific symptom complex or that specific symptom complex, you're not going to have the impact that you will have by working together. And I say this as a result of work that I've done in other fields where people have difficulty getting resources. Originally HIV/AIDS and autism, and obviously chronic fatigue is an example where, again, I think it's very important for you to be together so that you can get the resources that are required to find solutions to problems. The thrust of my own research since the early 1980s has been the discovery of microbes, bacteria, viruses, fungi that are implicated in causing diseases. They may or may not be present at the time the disease is manifest, but they serve as triggers for the immune system to cause the disease that's required. During the course of my presentation today, which is somewhat will be obscure to some of you, but I think obviously this is a very intelligent audience. You wouldn't be here. I hope to be able to have an opportunity to at least illuminate some of the ways in which one can begin to address these sorts of issues as triggers. Obviously, I don't do much more than simply serve as the front person for the work that's done by many people, and those people are listed here. And the people who are most critical, I think, in addressing the challenges of CFS in our group are a variety of clinicians whose names will be known to you. They're all based in the U.S. The people who participated in the original XMRV PMLD study, and of course my colleagues at Columbia who are focused on very many aspects, immunological, microbiological, and others. Now, we had a tragedy a few years ago, 2010, 2011. It was a setback temporarily when XMRV and PMLD were developed and promoted and such. But in retrospect, there's a silver lining to that cloud, and that it drew the attention of many people who are eminent in science. When you think about this, I don't think our organizations would be as far along as they are, or they're not far as they need to be, were it not for the interventions, the spotlight that was shown on the disease as a result of the reputed discovery of these agents that might be causative agents. I think it's worthwhile talking about the way in which scientists like to think about proving a relationship between finding anything and the disease, and there are many different ways in which this is done. The first person to really address this in a coherent way was Robert Koch in the 1890s in thinking about tuberculosis. And the principles that were put forward at that point, that they had to find a bacterium or a virus, at that point really didn't know much about viruses, but presumably viruses as well. In every case of disease, it had to be specific, and you had to be able to grow in the laboratory and put it back into an animal and reproduce the signs of disease. But it's going to be very difficult to develop an animal model, I think, for CFS-MEs, particularly when we're talking about things like cognitive dysfunction, or as many of the lice like to call it brain fog. I don't know how long we do that in a mouse. So we clearly can't wait for the development of such a model. So we have to have alternative approaches. In addition, there are instances where we can't really grow, a virus, in a cell line, or a bacterium in a cell line. So what we need to do is to develop alternative strategies for identifying infectious agents. And for the most part, those are genetic, meaning molecular biological. We identify sequences, and we look for relationships between those sequences and other sequences that are already known to represent infectious agents. But not everyone who's exposed to an infectious agent is going to come down with that particular disease. There's other host susceptibility factors, and this is why genetics is so critical too. So when we typically begin this sort of work, we make some sort of a link between the presence of bacterium or virus, because we're able to culture it. We're able to find the genetic sequence, we're able to find antibodies to it. We see it by electron microscopy or by light microscopy, and that's where the search really begins. If we then find that there are many people with the same agent, then that gives us more compelling proof that there's a relationship. If we show that there's a response to it, what we call an antibody response to it, that's indicative of recent infection, that's also very helpful. And if we have precedent for a similar model with a different virus, for example, or a different bacterium that's related to what we find in the human, that's also very helpful. But again, we don't have any such opportunities, I think, with CFS-ME because there is no really good animal model for the cognitive dysfunction and the depression and all the other things that are associated with this disorder. If, however, we can find a drug or a vaccine or something that mitigates disease or prevents disease upon exposure to whatever it is that's the offending agent, then that would give us clear evidence and it would also lead us to some sort of treatment or preventative. And that, of course, is what we're all seeking to do. We're trying to find out why people become ill, what sustains that illness, and what we can do to mitigate or prevent it. Now, I just want to talk about some models by which we can begin to understand ways in which infectious agents can cause dysfunction. I'm also going to give you some examples of intoxications, things that are common in the environment, but which may also, in a genetically susceptible human, result in some sort of disease that would be like the ones that concern us here. 7 years ago, 30 years ago, remarkably, these two scientists who are no longer with us, Rudy Rock and Marie Kaprowski, were the first really to describe antibodies to a virus that they thought was the cause of bipolar disorder, or what was then known as manic depression. And in what was then the first time that anybody used molecular methods to identify an agent, we identified nucleic acids that were specific to that particular virus, and we reasoned that once we had those in place, it would be relatively straightforward to figure out whether or not it did or did not cause disease. In fact, it took over 20 years to completely eliminate any possibility that this agent was associated with disease. And during that time, there were all sorts of things that people did which were dangerous, misguided, and there were all sorts of issues that we want to avoid when we're thinking about this disorder. Whether we're talking about Ebola or we're talking about CFS-ME, one of the first rules in medicine is prima norma care, which means first cause no harm. So we want to make certain that we know what we're doing so that we talk about clinical trials and anything else that we have the best possible evidence, that we do studies in a way that's coherent and safe so that we get to the answers quickly. And that's critical for establishing credibility within the field as well. You want to make certain that you have the best scientists in the world focused on this set of disorders. And to the extent that we're perceived as not pursuing credible science, we are not going to recruit those people and we are not going to make progress. So we have to be very serious about this as we are and try to make certain that the science is the best. And the organizations represented here today, I think are evidence of the sort of effort that's not being invested in that way. I first became interested in chronic fatigue syndrome, and I'm not known by that name, when I was contacted by Dan Peterson in the early 1980s to evaluate some of the first patients who were found in Yukon Village. And my next engagement was in the late 1990s when I began working with Brigitte Evengard, who's no longer in this field, but made many contributions to it, in looking at the potential for this particular virus called Lyme disease virus that had originally been implicated in these other disorders. We looked at that very diligently for a period of 2 to 3 years. We found no evidence whatsoever that this virus was implicated, despite the fact that 2 groups in Japan had said that it caused 50% of chronic fatigue in Japan. There were all sorts of problems with the assays they did. Very similar to what later happened with XMRV. But what I did say, because many people at that point were talking about psychosomatic issues and such, is I mentioned, and I said this in the discussion, I said, right, serum immunoreactivity to M.E proteins observed in Swedish CFS patients may reflect infections related to microbial agents that induce cross-reactivity with PDB protein. The fact that there is binding to a number of other proteins indicates polyclonal B cell activation. So I said, one thing is very clear. These people have some sort of immunological disorder. And we wrote this paper in 1997. It took 2 years to get it published. because there was a lot of concern about, I don't know, the importance of CFS-ME in those days. But even then, as I said, really back in the 1990s, I was convinced that this was a biologically based problem. It was not psychological, not to say that there aren't interactions between the brain and the body. The fact is, it was bona fide. And then, of course, we then get back to the XMRV PMLD story, which is fast forward, more than 10 years, and I began working with these very distinguished group of colleagues here. And I'm happy to talk about the XMRV PMLD story if anybody wants to do so, but I think we can lay that to rest.